Today we will discuss about a review paper that is published in the Nature, where they mention about this STUB1 gene that derives the self-renewal or differentiation behavior. So this Nature paper is based on the two independent papers that are published that explain the function of this STUB1 with respect to self-renewal or differentiation of the cells. So the idea is that this STUB1 this SATB1 this SATB1 levels, when they are high in any kind of cells, then it will make the cells to behave like a self-renewal state, and cells will remain in the same state until these SATB1 levels are high. However, when the SATB1 levels are low, then these cells will behave. Then these cells will behave towards the proliferations or differentiations. And the examples that this review paper shown is about the T cells. So in the T cells, when these SATB1 levels are high, then they will remain like a quiescent T cells, quiescent CD8 plus T cells, and they will continue to self-renew itself. However, when the SATB1 levels are low, then they will tend to differentiate towards effector like T cells or this terminal T CD8 plus cells. So this is one of the examples that they have shown. So what are the applications of this informations that has been published in the paper? One of the implications is about in the tumors. The tumors, it has been seen that SATB1 levels are usually high, and that keeps the tumors to remain in its self-renewal state. However, if in the case where SATB1 levels are low in the tumors, it has been seen that it is more prone towards these immune checkpoint blockade therapies, whereas when the SATB1 levels are high, then they are less prone towards these SATB1 therapies. So one usually want ST1 levels are low in the tumors so that they can be diagnosed by immunotherapeutic approaches. On the other hand, where we want these ST1 levels to be high, we want these ST1 levels to be high in the memory T cells, because if the memory T cells has high SATB1 levels, then it will cause these memory T cells to last longer, as they will have more self-renewal properties, and this will helps the immune system to maintain a immune response against a already identified known disease for a longer duration of time. So in that applications, we want the SATB1 levels to be high. However, in the case of tumors, we want SATB1 levels to be low. And as I have already told you, the examples that in the case of immune checkpoint blockade therapies, which usually block this PD-1 and CTLA-4 ligands on the T cells or on the immune cells, their efficiencies against the tumors is shown to be further accelerated when the SATB1 levels are low within the tumors. And also in terms of this differentiations of the T cells, so when the ST1 levels are low, then in the case of this immune cells, then they will more prone to make this effector like T cells, and this effector like T cells has the immune suppression properties. So in the immune systems, it is not desirable to have this low SAT1 levels. And one more examples that I have shown is about the memory T cells. It is mentioned in the paper, is about the memory T cells, where it is always good to have the longer life of memory T cells, because then they will be able to target the already identified disease for a longer duration of time. So I hope you understood the new finding that has been published in the Nature paper and the review papers, which is just compiles the results of those two independent research. That SATB1 derives self-renewal or differentiation behavior. It's not like it's the only one that derives, but it has a major contribution towards that. So that's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.